Welcome to the supplementary animation for our manuscript, Horizontal Antimicrobial Resistance Transfer Drives Epidemics of Multiple Shigella Species. What this study really does is document for the first time how the horizontal transfer of antimicrobial resistance has real-time impacts in determining pathogen epidemics. At the bottom of this slide, you can see the 3 epidemics that we were working with in England. They're all outbreaks of different types of the globally important diarrheal pathogen Shigella that were transmitting locally in England. To explore the outbreaks more fully and to look at the impact of antimicrobial resistance on the outbreaks, we complemented the epidemiological information from cases with analysis of whole genome sequences of isolates from those cases. Now, above the epidemic curves, what you can see is 3 whole genome phylogenies for the 3 different types of Shigella. For each type of Shigella, sublineages that we know were circulating in the same transmission network, for more details you can read the paper, are shown in colour. And these transmission sublineages were all associated with resistance to one particular type of antibiotic, azithromycin. What we found in our analyses early on is that a lot of these co-circulating isolates carried azithromycin resistance on the same plasmid. So by pulling out the plasmid sequences from all the 3 different types of Shigella, we could construct a separate phylogeny of the plasmid, and that's what you see here on the right. Now, by comparing the phylogenetic positions of genome sequences with the phylogenetic positions of the plasmid sequences over time, we can see what the real epidemiological impact of the horizontal transfer of the plasmid was on these disease epidemics at the population level. We sampled from 2008 onward because this was when the 3 types of Shigella were not considered epidemic in England. In 2009, we started to see a rise in the yellow type of Shigella, Shigella flexneri 3a. We think this was attributable to behavioural change in the community, as well as maybe other factors that we don't know about yet. This was also the year that we first observed the plasmid in the blue type of Shigella, Shigella sonni. In 2010, the plasmid moved into Shigella flexneri 3a, and you can see from the graph below that it starts to enhance the epidemic of that pathogen, with an increase in case numbers. Then, the plasmid appeared to circulate and diversify in this pathogen, because by 2011, we could see 3 distinct sublineages of Shigella flexneri 3a associated with 3 distinct sublineages of the plasmid. In 2012, we then saw the plasmid move into another sublineage of Shigella sonni, and also into the orange type of Shigella, Shigella flexneri 2a, where you can see that it starts to trigger an epidemic increase in that pathogen. In 2013, we see the plasmid move into yet another sublineage of Shigella sonni, and in 2014, the epidemics of Shigella sonni and Shigella flexneri 2a are well underway. Most of our sampling stopped at the end of 2014, but some extra data that we had from 2015 showed that the plasmid moved one more time into Shigella sonni. And here's the full data set you can see, showing the individual lines connecting up individual isolate genomes with their plasmid sequences. So what we've really been able to observe in this study is that the horizontal movement of antimicrobial resistance directly contributes to the epidemiological outcome of important pathogens. This means that in all areas of antimicrobial resistance research and public health management and surveillance, we need to be understanding our pathogens at this subgenomic level in order to understand the epidemiology of antimicrobial resistance. So thanks for watching our animation. Please check out the full paper in Nature Communications and find us at our respective institutions and online. Thank you.